this fourth joint webinar of Cost Action Atheronet and Marie Curie Staff Exchange Program Cardioscope. Today we'll discuss about animal models in atherosclerosis research. So these are the topics of the webinar. First, I will do a brief historical account just to remind everybody, and especially the junior colleagues, about the important role that rabbits play in atherosclerosis research, because they were the first animal models that were used. Then we'll discuss about more extensively about mice, which are the prototypical animal models for preclinical atherosclerosis studies. We'll explain the different types of mouse models, genetic models, nutritional models, and surgical models. We'll focus a little bit more on the genetically manipulated mouse models, because they are the more abundant and most widely used, which are the knockout, transgenic, knock-ins, and AAV-mediated, let's say, CRISPR-Cas9 models. We will briefly present the most widely used mouse models. And we will finish with some alternative animal models of atherosclerosis, such as rats, rabbits, hamsters, pigs, and zebrafish. And we'll end with the summary and the key points of the webinar. So as a brief historical account, rabbits were the first animal models of atherosclerosis research. And here you can see the two pioneers in this field. Alexander Ignatowski, who in 1908 fed rabbits with a diet enriched in animal proteins, milk, meat, and eggs, and observed intimal lesions with large clear cells, now they are called foam cell accumulation in the aorta. And in the decade of 1910s, Nikolai Nikolaevich Anitschko used a cholesterol diet dissolved in vegetable oil to produce aortic atherosclerosis in rabbits, similar to those seen in humans, and proposed a causal role of cholesterol in atherosclerosis. So these pioneering studies provided the first experimental evidence and basis for the establishment for the lipid hypothesis of atherosclerosis. And several significant milestones in the history of the study of atherosclerosis are based on tremendous breakthroughs and insights using rabbit models. We'll come back to the rabbits later when we discuss about alternative models. Just to show you this slide, which shows the total paper number published each year using rabbits or mice for atherosclerosis research between 1970 and 2014. And you can see that since 1970 up to approximately 2000, rabbits were the predominant models of atherosclerosis research. At some point in 1992 and later on, the rabbits were replaced by mice. And in this decade here, between 1990 and 2000, this was actually a big bang in atherosclerosis research, because there were tremendous breakthroughs in the generation of genetically manipulated mouse models, as we'll discuss later. This slide shows the yearly publications of different animal models used in atherosclerosis research between 2000 and 2021, and you can clearly see that the two most frequently used models are the ApoE knockout mice, shown with the blue line, and the LDL receptor knockout mice, shown in the red line, with the red line. But also we have rats, shown in green here. We have rabbits, again, in this purple line, and we also have pigs, zebrafish, and non-human primates. But it's clear, actually, it's clear from this slide that the mice and the genetically manipulated mice, these two models, the ApoE knockout and the LDL receptor knockout mice, predominated the field. Most of the studies were done using these two mouse models that we'll discuss later. So the mouse is the prototypical animal model for preclinical atherosclerosis studies. And despite the significant differences between mice and humans regarding their lipid profiles, the types and the anatomical sites of plaques and their composition, mice have been used extensively for atherosclerosis studies for the following reasons. They have a short life cycle. Their cost of maintenance is low. They produce large numbers of progeny many times a year. They are easy in genetic manipulation, and there are plenty of strains available with different genetic backgrounds. Now here, this slide compares the lipoprotein cholesterol distribution between species. So the mice are here. Here is the mouse. And we can see that most of the cholesterol is distributed in the HDL fraction and less cholesterol in the VLDL or LDL. And this is due to the absence of CETP in mice, as we discussed in the previous webinar, and we'll discuss again later. This is the lipid profile in humans. We can see that most of the cholesterol is in the LDL and less cholesterol in HDL or VLDL. And these are the lipid profiles of other animal models. And we can see that the animals that have a profile which is most close to human is the pig, the monkeys, and the cows. And the others are not very close to the human profile. However, you can make mice have, and definitely this profile of the mouse is not an atherogenic profile. You cannot easily generate atherosclerotic plaques in animals that have this lipid profile. However, you can make this profile more atherogenic if you do some genetic manipulation. And in the right-hand side, we can see the lipid profiles now of different mouse strains. This is the wild-type mice, the C57 Black 6J. Again, this lipid profile. But now, if you can, let's say, overexpress ApoB in these mice, and these are the transgenic ApoB mice. We'll discuss later how we can make transgenic mice, but these are the transgenic mice. You can see that this lipid profile is very close to humans, which is here. This is the lipid profile in humans. This is the transgenic ApoB mice. Also, these mice that express a gain of function mutation of PCSK9 has a lipid profile which is similar to humans, as similar to the LDL receptor knockout mice. These are the ApoE knockout mice. This profile is extremely atherogenic. You can see that most of the cholesterol is distributed in the LDL and VLDL region, and very little cholesterol in the HDL. And we can discuss later that these mice are the most atherogenic because you can have atherogenesis even in the absence of a high-fat diet, whereas in the other animal models, you need a high-fat diet to create atherosclerotic plaques. Now, how can you detect atheromatous plaques in animals? In mice, most of the plaques are formed in the aorta, and you can excise the aorta and do an aphasia analysis. You can stain with a dye that stains lipids, such as the Olered O. And this is an analysis of the aorta of ApoE knockout mice fed a chow diet, and this is an ApoE mouse with a Western type diet, and you can clearly see the development of atherosclerotic plaques throughout the aorta, but primarily in the aortic root and the aortic arch. You can also study the plaques better. You can do cross-sectioning of the aortic roots here, and you can stain with H&E just to do to watch the morphology of the aorta in this region. And you can stain with Olered O to detect the lipids, the accumulation of lipids. And also you can stain with MoMA2 in order to detect the macrophages, and the presence of macrophages indicates inflammation and an unstable plaque. Now, there are several types of preclinical models for atherosclerosis, and these are the three main types: the genetic models, the nutritional models, and the surgical models. And we will start with the genetic models, and we have several types of genetically manipulated models. We have the transgenic animals, that they express the gene under study simultaneously with the endogenous gene. The knockout animals which do not express the endogenous gene, and then in animals in which the endogenous gene has been replaced by the exogenous one, either the wild type or a mutated version of it, and the regulatory regions of the endogenous gene are preserved. So just a very brief description of each model. So these are the transgenic mice. In order to generate a transgenic mouse, you just take a fertilized egg before the fusion of the pronuclei, and you inject your gene of interest into the male pronucleus, which is the larger, and then you take this injected fertilized egg and you implant it into pseudopregnant mice, and you analyze the offspring for the presence of the transgene using genotyping, usually by PCR. It used to be Southern, now you do this by PCR. You just take a small piece of the tail of the mouse and you isolate DNA and do the genotyping using PCR with primers that correspond to your gene of interest. You can also overexpress a gene into an animal without transgenesis. You can use viruses as vectors. So this is an AAV. It's a vector that has been used extensively for long-term gene transfer and expression in animals, in various animals. We can see later that the AAVs have been used to generate animal models of different species, not only mice. So essentially, what you do is you take a plasmid DNA which contains the gene of interest, the transgene, and you package it into recombinant AAV particles. So these are the particles that carry the transgene, and you administer these particles into the host organism. That can be mouse or it can be rabbits or other species. Then by doing that, you transduce the host cells. So the recombinant AAV releases single-stranded DNA into the nucleus, and then the transfected cells express the transgenic protein. We'll see later that this system has been used to transfer the PCSK9 gene into mice, and that is sufficient to generate a very good mouse model of atherosclerosis. The knockout mice is generated by isolating a blastocyst, an embryo from a pregnant mouse. So you isolate the blastocyst and you take the embryonic stem cells from the inner cell mass, you culture them, and here in these Petri dishes, you do the gene targeting. That is, you destroy your gene of interest. You replace it by, let's say, by another gene, usually a marker gene. And then these are the genetically modified embryonic stem cells. You take them, you inject them into the cavity of a blastocyst from a different mouse strain, and you reimplant it into a foster mother. And then what you get from here is chimeric mice. And in order to identify the mice that contain in heterozygous form the knockout, the genetic manipulation, in all nucleated cells, you breed it with a wild type mice. And you take the protein that do not express the gene in all cells. And this is a recent breakthrough in gene targeting in the gene targeting field. This is the CRISPR-Cas9 method, by which you very efficiently and very specifically you can mutagenize a gene of interest very specifically. So basically what we have here is a two-component system. One is a nuclease called Cas9, shown here, and an RNA component, which is the RNA guide that guides the Cas9 to the region of your gene that you want to mutagenize. And so when this complex of Cas9 and guide RNA binds to your DNA, the Cas9 creates a double-strand break. The base repair machinery repairs the break, but it does mistakes, and that's how you mutagenize your gene. It's a very efficient method. In order to use that in animals, you place both of these components into a single vector, usually AAV, as you can see here. So this is a CRISPR construct for genetic engineering. It has the guide RNA, it has the Cas9, and then you make AAV particles using this genome, and you transduce animals, cells or animals with this construct through viral infection. Now we continue with the nutritional models. In the nutritional models, you just feed the animals with a high-fat diet. I remind you that mice are very resistant to atherosclerosis, and they need, even the genetically manipulated mice are resistant, and they need to have a more atherogenic profile. And this atherogenic profile is generated using these types of diets. The most widely used diet is the Western type diet or high-fat diet shown here, which contains 21% total fat, 60% saturated fatty acids, 34% sucrose, and 0.2% cholesterol. If mice are resistant to these mice, then you increase the cholesterol, and you have the high cholesterol diet, which contains 1.25% cholesterol and 40% fat, or you can even go to 2% cholesterol. And for some mice, you need to have a more, let's say, aggressive but toxic diet, and this is called the pageant diet, which contains sodium cholate, which is toxic, but it's a more potent atherogenic diet. And finally, we have the surgical models. Here we use these surgical operations in order to induce atherosclerosis into arteries that they do not normally have atherosclerosis, such as the carotid arteries, for instance. Mice do not develop atherosclerotic plaques in the carotid arteries. They usually develop plaques in the aortas. So in order to have plaques in the carotid arteries, you need to have some surgical operations, and there are three main surgical operations. One is the complete carotid artery ligation. So you ligate the carotid artery using a suture. So you have a complete flow cessation here in the carotid artery, and this can cause a very drastic atherosclerotic plaque development. You can have decrease of lumen area by 80% in this carotid artery. You can have carotid artery stenosis. That is, you put a collar into the carotid artery, and you can disturb the hemodynamics in this area. So you can have oscillatory blood flow here, low shear stress here, and this is sufficient to develop plaques that have a decrease in the lumen area by 60 to 80%. And finally, you have the partial carotid artery ligation, where you don't have a complete ligation, but you have a partial. You just ligate the occipital artery, the internal carotid artery, the external, and only the superior thyroid artery remains open. And this is sufficient to cause low and oscillatory blood flow, and that causes atherosclerosis, including intraplaque hemorrhage. So now now we have completed the presentation of the different models, different systems that we can use to develop these models, and we can proceed with a brief presentation of the most widely used mouse models. And most of the widely used models of atherosclerosis research contain mutations in genes which participate in the LDL pathway shown here. So I showed this slide also in my previous webinar when we discussed about the lipoprotein pathways. So this is the pathway of low-density lipoprotein. Very briefly, the liver secretes ApoB, which is packaged with lipids with the help of MTP, and this forms the VLDL particles from the liver. The VLDL particles are hydrolyzed by lipases, such as lipoprotein lipase or hepatic lipase. These free fatty acids are transferred to peripheral tissues, and the remnants are called IDL and eventually LDL, and these remnants are cleared with the use of ApoE. Now, of course, ApoB is very important for this pathway, not only for the formation of these lipoprotein particles, but also for their clearance from the circulation, because ApoB is the ligand for the LDL receptor. ApoE is also the ligand of the LDL receptor. When you don't have ApoB, then LDL accumulates in the circulation. It can be converted to oxidized LDL, and which can be taken up by the macrophages, and we can have the formation of foam cells and atherosclerotic plaques. When you don't have ApoE, you don't have the clearance of the remnants. Again, it creates a very atherogenic profile, but most of the cholesterol is distributed in the remnants, not in the LDL, but in the VLDL and IDL particles. The LDL receptor, of course, is very important. When the LDL receptor is mutated, then you don't have the clearance of these particles, and they accumulate in the plasma, and they can cause atherosclerosis. And finally, we have PCSK9, which is a protein that is expressed primarily in the liver, and it has been shown to bind to the LDL receptor and degrade it. So when you have high expression of PCSK9, this destroys all the LDL receptor, and again you have the same phenotype, that is a very atherogenic lipoprotein profile. So the mice I'm going to describe you now, they have mutations either in ApoE or in PCSK9 or in the LDL receptor. These are the genes that are most widely used for the generation of animal models. But again, overexpression of ApoB is also atherogenic because it leads to the formation of more VLDL particles and eventually more LDL particles. And we'll start with the ApoE knockout mice. This is the first model. So here we have this disruption of the ApoE gene. This is the lipid profile. Plasma cholesterol in chow diet is between 400 and 600 milligram per deciliter, but on a Western type diet can go up to 1,000 milligram per deciliter. As we discussed, most of the cholesterol is in the VLDL and the LDL and less in the HDL particles. This is a very atherogenic profile. These are the plaque distribution characteristics after the administration of a high-fat diet. But these mice can have atherosclerosis even without a high-fat diet, but the plaques are not so advanced. So we have fibrous plaques. And the advantages and limitations, the advantage is that it develops atherosclerosis even in the absence of a high-fat diet. But the limitations is that the lipid profile is not close to humans. The humans do not have such a profile. Most of the cholesterol is VLDL and less in HDL. ApoE also is expressed in macrophages and plays a role in inflammation. Here we don't have ApoE, so we have a problem, let's say, interpreting this data, because ApoE in macrophages also can protect from atherosclerosis, not only in plasma as a ligand for the LDL receptor. So it is a more complex model. And also we don't have a spontaneous plaque rupture, thrombosis, and other complications. So this is actually from the original paper from a group of Ian Breslow in the Rockefeller Institute. And you can see in this table that the total cholesterol and the triglycerides in these mice, in ApoE deficient mice. So here you have the wild-type mice, the heterozygous and homozygous in chow diet. And here you have, sorry, the heterozygous, the wild-type. These are the wild-type mice. These are the homozygous knockouts, and these are the heterozygous. And this is in the absence or in the presence of a Western-type diet. And you can see that even in chow diet, these mice have high levels of total cholesterol, up to 400 milligram per deciliter. But if you feed these mice a Western-type diet, these total cholesterol levels can go up to 1,800, as you can see here. The triglycerides do not differ very much. And this is a graph showing the lesion area in the wild-type mice or in the knockouts. And you can clearly see that even in mice that receive a chow diet, the knockout mice develop atherosclerotic lesions. which are more exacerbated when they receive a high-fat diet, a Western type diet, as you can see here. And most of the cholesterol is in the VLDL and LDL, and very little cholesterol is in the HDL region. The other model, most widely used model, is the LDL receptor knockout mouse. Here we have the disruption of the LDL receptor gene. Plasma cholesterol in chow diet is between 200 and 300. On the Western type diet, more than 1,000 milligram per deciliter. So this is a model that mimics FH, human FH. You'll see in the next slide that these mice also develop xanthomas as patients with FH. Most of the cholesterol is in the LDL region and less cholesterol in the VLDL. The HDL does not change. We have again fibrous plaques, but most of the plaques develop in the aortic arch and root, as you can see here. The advantages is that you have a more human-like lipid profile compared to the ApoE knockout mice, and you have functional ApoE in macrophages, so there is no impact on inflammation. But the development of lesions is less complex and requires a Western type diet, and again, we don't have a spontaneous plaque rupture, thrombosis, and complications. And this is the paper, the original paper from Goldstein and Brown from Dallas in 1994. Here you can see the cholesterol levels in wild-type mice, heterozygotes and homozygotes, in male and female, and you can see that the cholesterol increases only in the homozygotes and very much in the presence of a diet which is rich in cholesterol, 1.25% cholesterol. These are the xanthomas that these mice develop, similar to humans with FH, and this is the development of atherosclerosis in mice, knockout mice, in the presence of a high-fat diet. And most of the cholesterol is in the LDL region. The other mouse model is the ApoE3 Leiden that has been used by many groups. This contains a mutation. This mice is a transgenic mouse. It contains the human ApoE3 Leiden mutant. This is a mutation in ApoE which prevents binding to the LDL receptor. So in these mice were generated by using a transgenic cassette which contains the ApoE3 Leiden along with the ApoC1 gene, which is very close to ApoE in the locus, the ApoE and ApoE C1, C2, C4 locus. So it expresses a mutant human ApoE. The plasma cholesterol in chow diet is between 100 and 200, and in the Western type diet, more than 1,000, very, very similar to the previous model that we have here, the LDL receptor knockout mice. It has most of the cholesterol is in VLDL and LDL and HDL, only in the presence of Western type diet. Again, we have plaques formed in the aortic root. We have functional ApoE, and the limitations are the same as the previous models. Now, this model can become better, especially if you want to study metabolic syndrome, if you take these mice and you cross them with CETP, transgenic mice. So these mice express the CETP, and I would like to remind you here that the CETP is the plasma enzyme that transfers cholesterol esters from HDL to the LDL, in order to take them back to the liver for secretion. So when you overexpress CETP, you have exaggerated transfer of cholesterol esters to the VLDL, LDL, so you have less HDL. So by overexpressing, and mice lack CETP, they don't have CETP. So if you overexpress CETP, immediately you see that the HDL levels become lower. So this is the lipid profile of wild-type mice. Again, most of the cholesterol is in the HDL fraction. If you overexpress CETP, the cholesterol HDL fraction is reduced, and most of the cholesterol now is distributed to the VLDL-LDL region. So this is the profile of the ApoE3 latent CETP mice, which is very close to human, and this is the profile of the ApoE3 latent mice, which is not very close to human. Both of these profiles are atherogenic. If you take these mice and you feed them a high-fat diet, they develop atherosclerosis, as you can see here, even severe lesions. And also in the liver, you have steatohepatitis. And these mice, they are very good for pharmacology because they respond to drugs such as statins or CETP inhibitors. And just very briefly, I would like to show you some data from our lab. About 4 years ago, we published a paper in which we studied the effect of high-fat diet on global gene expression changes in the liver, just to show you how these mice can be used. So these are ApoE3 latent CETP mice. We divided them into 2 groups. In one group, we gave high-fat diet for different time periods, 4, 8, and 12 weeks. And the other group, we gave low-fat diet as a control. And then at the end, we sacrificed the mice, and we evaluated them clinically for blood parameters and also for circulating microRNAs and histopathology. And from liver sections, we performed transcriptomic analysis using expression arrays. This is the blood parameters of these mice. So these mice, especially the mice that receive the high-fat diet, they are glucose intolerant, as you can see here. They are insulin resistant. They have hyperinsulinemia, and they develop hepatic steatosis. So these mice with a high-fat diet, they have all the characteristics of the metabolic syndrome, and then you can use these mice in order to study atherosclerosis, or you can do other things such as study the transcriptome in the liver, as we did, and we identified changes in several key genes that could explain the phenotype in these mice, such as overexpression of PPAR gamma, reduction of leptin receptor, which can lead to obesity, and also we have increased expression of some genes which are important for the lipid droplet formation, and that can also contribute to the development of hepatic steatosis. And the next model is a mice which express the PCSK9 using the adeno-associated viruses. So these mice have plasma cholesterol on chow diet about 300 milligrams per deciliter, more than 1,000 on Western type diet. The profile is very close to the LDL receptor deficient, and these mice are very closely mimic the LDL receptor deficient mice. The limitations and all the characteristics are the same. So PCSK9 is a gene which binds to the LDL receptor and degrades it. So when we have gain of function mutations, we have increased PCSK9 activity, increased affinity for the LDL receptor, increased LDL receptor degradation, increased LDL cholesterol in the plasma, and increased risk for cardiovascular disease. In humans, these mutations cause FH. And one of the mutations that causes FH in humans and has been used in mice is this aspartic 374 tyrosine mutation, the D374Y. This is a gain of function mutation. It generates a PCSK9 which is more potent than the wild type protein. And as you can see here, this is from a paper published approximately 7 years ago, in which they did a single injection of AAV PCSK9 mutant in mice. They saw that this treatment induced atherosclerosis in high-fat, high-cholesterol fat C57 Black 6 mice. So all these images are from the aortic sinus and the aortic arch, staining with oily red dough, and you can see the development of atherosclerosis. So these are the mice that received the PCSK9 AAV, and these are the LDL receptor knockout mice. And you can see that these mice have very similar morphologies. So the the pathology of these mice are very, very similar. So this is a very convenient method. Instead of, let's say, generating LDL receptor knockout mice, you can actually mimic this LDL receptor deficiency in any model that you want. You can combine the LDL receptor deficiency caused by the PCSK9 overexpression with other genetic defects. You can have combined models of atherosclerosis. So this is a very nice model, very convenient. And also we have mice which can develop plaques which are more unstable and more prone to rupture and thrombosis. And these are mice that essentially these are apoE-knockout mice which can be crossed with mice that contain a mutation in the fibrillin-1 gene, this specific mutation. These mice that express the fibrillin-1 mutant, they have mutation in fibrillin, which is an essential component of elastic fibers. So the aortas are not so rigid, and they can have problems in the connective tissue in the aortas. So in mice that have these mutations, you can easily have plaque rupture and neovascularization and hemorrhage. Also, you have MI and stroke and sudden death, which these are characteristics that are due to the instability of the elastic fibers here that the other models that we described, they do not have. So this is a very good model if you want to study the vulnerable plaques and let's say hemorrhage and neovascularization in mice. So this is an image from a paper published in 2009 in which they crossed the apoE deficient mice with fibrillin-1 mutant mice, and they saw that you have in the aortas, you have a disruption of the fibrous cap, and they found the presence of red blood cells, which are indicative of hemorrhage. And finally, we have another mouse model that was developed during the past decade, which is a good model if you want to study coronary atherosclerosis. You remember from the previous slides that all the other mice that we described, they do not develop atherosclerosis in the coronary arteries. They develop only in the aortas. Here, in this mouse model, you can have atherosclerosis in the coronary artery. And this is a model that you can create if you cross apoE knockout mice with SRB1 knockout mice. SRB1 is the scavenger receptor that binds HDL. It's the HDL receptor. So you have a double knockout mice here, either apoE knockout mice, SRB1 knockout mice, or LDL receptor knockout mice, SRB1 knockout double knockout mice. So this crossing here is sufficient to induce atherosclerosis in the coronary artery. As you can see here, you can have even 100% occlusion in the coronary artery. And you can also have myocardial fibrosis, as shown here in the SRB1 apoE double knockout mice, 3 weeks. So the advantages of these SRB1 compound deficient mice are they develop large and complex atherosclerotic plaques. They develop coronary artery atherosclerosis characterized by accumulation of platelets and myocardial fibrosis, and they are good mimics of human coronary atherosclerosis. So one of the few models that have been generated and they develop coronary atherosclerosis. apoE knockout mice is a model of this. It's a model that does not have HDL. We all know that HDL is the good lipoprotein that protects from atherosclerosis. And when you have very low levels of HDL, you are not protected and you develop atherosclerosis, at least in humans. In mouse, this is not what is happening. These mice, even though they contain very low levels of HDL cholesterol, as you can see here, these are wild-type mice, and these are apoE1 knockout mice. They have some HDL, but this HDL does not contain ApoE1, contains other apolipoproteins, primarily ApoE. So these mice do not develop atherosclerosis even after a 14-week feeding with a very potent diet such as the Pageant diet that I showed you. So these are the wild-type mice after 14 weeks of Pageant diet, very little atherosclerosis. This is quantitated here. These are the ApoE1 knockout mice. Again, very few, very small atherosclerotic plaques compared to wild type. So these mice are very resistant. Even though they do not have HDL, they are very resistant to atherosclerosis, even in the presence of a very potent high-fat diet. But to our surprise, we found that you can increase atherosclerosis in these mice if you overexpress ApoE1 mutant forms, such as the ApoE1 PISA and Finland, which are two mutant forms that create HDL deficiency because they do not activate LCAT. So as you can see here, you can have increased atherosclerosis in these mice. So in other words, this is one of the few studies in which atherosclerosis could be developed, could be generated, and exacerbated in a mouse model that is not genetically manipulated by mutagenizing the ApoE or the LDL receptor. So you can have atherosclerosis, but even in mice that do not have mutations in LDL or ApoE, but the atherosclerotic plaques are very small. So this is the mice that we generated for this project. And in the last part of my presentation, I will show you some alternative animal models for the study of atherosclerosis. Of course, we have the rats, which are bigger. They have bigger size compared to mice, but they are more difficult to genetically manipulate using the embryonic stem cell technology. However, recently, the investigators, they have achieved the generation of LDL receptor knockout and ApoE knockout and double knockout rats. And they found that these rats develop atherosclerosis, but only in the presence of a very drastic diet, such as the Pageant diet, for a very long period of time, approximately 50 to 64 weeks. As you can see here, they develop atherosclerosis, but they need to receive diet for extended periods of time, which is something that is not practical to wait for such a long period of time to have animals with atherosclerosis. However, they are bigger, and you can see the plaques better because they have larger arteries. Rabbits have similar lipoprotein metabolism to humans because they express CETP. They grow easily and in big numbers with low cost. The most widely used rabbit is the Watanabe heritable hypercholesterolemic rabbit, which brings a mutation in the LDL, natural mutation in the LDL receptor gene, and they develop spontaneous atherosclerosis. We have also recently generated ApoE knockout rabbits, which develop atherosclerosis only in the presence of a high-fat diet. And this is a small table comparing humans with rabbits and mice in terms of lesions in the aorta or the coronary artery or cerebral lesions. And you can see that rabbits, they also develop coronary lesions similar to humans, but mice cannot develop lesions in the coronary arteries. The Syrian hamsters, they have similar lipoprotein metabolism to humans. They also express CETP. And recently, using the CRISPR-Cas9 technology, we succeeded in obtaining LDL receptor knockout hamsters, which develop atherosclerosis in the presence of a high-fat diet for 12 weeks, and they also develop a myocardial infarction. So here is some images from the aortic root, the lesion areas. So these are the wild-type hamsters, and these are the LDL receptor. heterozygotes that develop atherosclerosis, even in the coronary artery, as you can see here, similar to humans. The zebrafish, they are easy of breeding and maintenance. Genome editing is possible. Environmental impact is enough to induce hyperlipidemia without any genetic manipulation. Embryos and larvae are transparent, which enables in vivo non-invasive monitoring, and they express CETP, PCSK9, and other lipid metabolism genes. You can see here, it's very easy to stain, let's say, for lipid deposits in the ventral vein in these animals. When they develop these lipid deposits, when they receive a high cholesterol diet. And finally, we have the pigs, the larger animals, which are expensive and difficult to handle. They are difficult to genetically manipulate. Recently, PCSK9 gain of function mutant expression Yucatan mini pigs. This is different, different than the normal pigs. They have been created and show severe hypercholesterolemia and development of progressive atherosclerotic lesions. And they have similarities with human plaques, coronary atherosclerosis, and neovascularization. So finally, now that we have presented all the different models, let's briefly summarize what are the factors that we need to consider before we select a mouse model for atherosclerosis research. First of all, we need these are factors that we need to consider. Of course, we need to select the appropriate mouse strain. I hope that you appreciated that not all of the strains are the same. Some strains they develop atherosclerosis very easily. Some strains are very resistant, and we need to feed them a very potent high-fat diet for longer periods of time. Some strains develop stable plaques. Some strains develop unstable plaques. So the selection of the mouse strain is very critical. We need to consider also sex differences, and actually now it's better to use both sexes, both male and female, to study atherosclerosis, not only one of these. The diet and the duration of the diet is very important. Some mice are resistant, let's say, to a simple high-fat diet. We need to increase the concentration of cholesterol, and we also, for some mice, we need to give it for longer periods of time, such as in rats, for instance. Age also is very important. Usually we use young mice for atherosclerosis research, but in humans, atherosclerosis does not develop early. It develops, let's say, at later stages. So perhaps when we use mice that are not so young, it's better when we can find different things in atherosclerosis. And of course, the circadian rhythm plays an important role. There are many studies that shows that this is critical, especially when we need to decide when to do the nutritional intervention or when we do the surgical operation, in what time of the day. And these are the characteristics of human plaques that animal models should recapitulate. The lipid profile should be very similar to humans if we're going to have a good animal model that mimics human atherosclerosis. We need to have a model that has advanced stages of atherosclerosis, plaque necrosis, atherothrombosis, intraplaque hemorrhage, neoangiogenesis, and calcification. And we discussed some models that have these features. The location of the plaque is important. In humans, we have plaques in the arteries, not in the aortas. We have some models that have coronary atherosclerosis. Plaque regression, some models are ideal to study that. We can study plaque regression in mouse models, and also plaque rupture. There are few models that have unstable plaques, such as the apoE-fibrillin-1 model. And this is the final slide. This is a summary and the key points. Rabbits were the first animal models that were used for atherosclerosis research, but were replaced by mice after the discovery of genome modification techniques. The mouse is still the prototypical animal model for preclinical atherosclerosis studies due to their short life cycle, low cost of maintenance, easy genetic manipulation. There are plenty of mouse strains available, but the most widely used models are the APOE knockout mice and the LDL receptor knockout mice. These mice have been used as base models for additional genetic, dietary, or surgical manipulations. CRISPR-Cas9 technology has allowed the genome manipulation of additional animal models such as rats, rabbits, or hamsters. And finally, despite the advances in animal modeling, a preclinical in vivo model that accurately mimics human atherosclerosis physiology is still missing. And with that message, I would like to finish, and I would like to thank you all for your attention, and I would be happy to answer any of your questions. Thank you very much.
Thank you so much, Dimitris, for this very comprehensive overview of all the possibilities when it comes to preclinical models out there. I know that you are currently preparing also the paper regarding this issue, so I think everything is really up to date. We have a question already from the audience. Let me just try to go. Okay, so from Eliana Ibrahimi. Some studies report that atherosclerosis could be much more related to glucose metabolism disorders and high-carb diets than with high-fat diet. What's your thoughts on that?
Well, yes, actually, a high carbohydrate diet can cause atherosclerosis, but I don't think that this is a very useful model to study, let's say, lesion development. They're not so much used, these mice. So most of the mice that are used for atherosclerosis are mice that contain this atherogenic lipid profile. Of course, the insulin resistance and hyperglycemia is observed in the mice that receive a high-fat diet. Of course, you have that. So this is a component that contributes to atherosclerosis, but without an atherogenic diet contributed by, let's say, a diet which is rich in high fat, I mean, it's very difficult, especially in mice, to have atherosclerosis.
Thank you. We have a question from Soner. He can. I sent you the request to unmute, so then you can be unmuted and then ask question personally. Thank you.
Yeah. Good evening, Dimitris. It was an excellent presentation.
Thank you.
Thank you. It was very educational. My question is about wild type mice. Do they develop atherosclerosis, or if they do, when do they develop?
They don't. They don't. Wild type mice, they do not develop atherosclerosis. I saw a slide in which we used wild type mice and we gave them a very potent diet, the pageant diet for 14 weeks, and the plaques are very, very small. They're extremely small. So wild type mice are very resistant, and that's why we genetically manipulate them in order to have a more atherogenic profile. They're very resistant because their profile is not atherogenic. They have high levels of HDL cholesterol, which does not form atherosclerotic plaques. In order to have atherosclerotic plaques, you need to have high levels of LDL, not HDL.
Okay, so regardless of their age, like 2-3 years, it doesn't matter. They're resistant.
They are resistant. Even old mice are resistant to atherosclerosis. Yes.
Okay. Thank you.
Sure.
Next question from Johannes. I'm sending you the request to unmute so you can now.
Hello, can you hear me?
Yes.
Yes.
Okay. Thank you very much, Dimitris, for this nice talk. Unfortunately, I could step in just later, so I missed the first part.
That's okay.
Maybe you mentioned it in the first part, but when you listen to Peter Libby, he actually says that the traditional atherosclerotic plaque is not seen that much anymore in the clinics because of the widespread use of statins, so that you don't see this kind of fatty streak plaques anymore, but you see more fibrous plaques. And
Yes.
the question is whether the mouse models with ApoE knockout and the LDL receptor knockout still being the most frequently used models, whether they reflect the clinical situation sufficiently well.
No, they don't. No, they don't. No, they are much different than the human plaques. That's for sure. The plaques are very stable. They don't have rupture. They don't have hemorrhages. So they are just stable. They mimic the stable atherosclerotic plaques in humans. So these are not good models if you want to study plaque rupture or thrombosis. The only good models are the SLB1 ApoE knockout mice and the ApoE, the fibrillin-1 gene knockout mice. These are the right now. These are the best models to study unstable plaques, not the other ones.
Okay, thank you.
Sure.
We have another question from the audience in the chat. The question from Martin, I think. He is asking which model of atherosclerosis regarding the influencing the inflammation. So which model would be good to study inflammatory pathways in plaques?
Again, these models, they develop, they have inflammation, but the inflammation is not the critical factor here. In all these models that we described, the most critical factor is the atherogenic profile, not inflammation. If you want to study the role of inflammation, then you need to have other models that you can induce inflammation in these mice. We have actually some work in progress showing that atherosclerosis can be exaggerated by chronic inflammatory diseases such as let's say, rheumatoid arthritis. In all these models, you can see inflammation, you can see macrophages in the plaques, but you don't see extensive inflammation from the pictures that you obtain.
Johannes replied that they have a model on that. So if somebody's interested, they can contact Johannes.
Okay, great.
Yeah, perfect. Paolo?
Okay, thank you very much, Dimitris. Now, I've not been working with animals since some time, but I work with cells and humans. But now, after this seminar, this webinar, I guess all of us would like to work with animals.
Good.
Because you excited all of us with all these opportunities, possibilities, and so on. So I have a general question. How is the timing of getting a proper lesion? I mean, whatever lesion each animal can give, but what is the time frame within you can get it? Because also this is also experimentally important, also economically important.
Yes. Well, it's not the same for every animal. Some animals develop atherosclerosis very easily, such as the ApoE knockout mice. In 8 weeks, you can start having plaques, good plaques. So you can, let's say, take these plaques and do some research. If you want more advanced plaques, you have to wait. But in most mice, atherosclerosis develops after at least 16 to 18 weeks of high-fat diet treatment. So you need to wait several months. These experiments are not fast. I mean, you need to wait. In all these atherosclerosis experiments, you need to have several months ahead of you in order to finish these experiments. It takes long. They are not easy experiments. They are expensive. The diets are expensive. We did not discuss about the financial aspect, but of course, mice are easy to generate and they are cheap to maintain, but the diets are expensive, all these high-fat diets, especially if you want to, let's say, to enrich them with different things. They become even more expensive. And because you need to give them for many, many months, so that increases the cost. But the time frame is approximately 16 to 20 weeks, let's say.
And sex and age of the, of course, usually maybe young adults are the optimal animals for these experiments. But
Yes.
is I mean, has anybody used older animals and see whether they develop different or faster plaques or?
Not really, not really. Most of the studies are done using young mice, 8 weeks or 10 weeks. They don't wait. I mean, very few studies. I don't think there are many differences.
Also, because if you buy older animals, they are quite expensive, so it can be.
Yes,
yeah.
yes. So there are very few data.
Yeah,
There's no clear answer to that.
Yeah,
Somebody used to do a serious study to see what is the effect of, let's say, aging in mice on atherosclerosis, because most of the studies are done using young mice.
Of course, but if you keep animals for 2 years, it's kind of very expensive.
Of course, and they have other diseases, aging diseases, for instance.
Yeah, sure, cancer also, yeah.
Yes.
Now, the other question is about male versus females. Are there many differences in mice or not?
There are some differences, but again, depending on the models. Some models have male mice are more, let's say, prone. In other models, females. They are not. There's not a uniform characteristic in these mice that, let's say, male are more resistant. This is not the case. Although you'd expect, if you compare with humans, that males are more susceptible. In mice, this is not the same. Females can also be very susceptible. So that's why if you don't want to have biases in your research, you need to use both sexes, both male and female, and see how they behave, not only males. So in our study, if you notice, we used only males, because it is known that this model, in this model, the apoethylite and CATP mice, the male mice are more prone. But this is not the case for all mice. So
the last comment is: can you tell us about your hamster model? Because seems like to be very interesting.
I don't use hamsters, actually. I don't have experience with hamsters.
Oh, I'm sorry. With the knockout, LDL knockout.
Yes, this is a published paper.
Okay.
I don't have experience with hamsters.
Is it something promising or not? In your
opinion? I think it is because they have a very good profile, lipoprotein profile, very close to humans, more close than mice. So it's a good model. It's a very good model, actually. But it's very difficult to genetically manipulate.
Okay.
I mean, just hamsters. But now with the CRISPR-Cas9, you can actually do manipulation in these mice as well, in these animals as well. But until now, it was difficult.
I think Loredan from Bucharest, they use the hamster model for some of the research.
In Bucharest, they use hamsters.
Yeah.
If somebody wants to collaborate and use hamsters, they can talk to the Bucharest people, the SCDP.
I also saw something that caught my attention, because more or less all others just asked the question that I was interested in, and also Paul underlined the sex differences, and this is something that was really, I think, unfortunate in the previous 30 years or even more, than most of the time, 90% of the time, male animals are used in this model, and now we have a lack of information about the animal models. What caught my attention in one of your slides, I think it was LDL double knockout mice, or maybe I might be wrong, but there was the information when you go to heterozygous, there is the rise in cholesterol to 150 milligrams, I think, and then when you go to homozygous, then males rise until
yes, I remember
130, and females go even more to 170 or something like that, which is quite strange. Is this something translatable to human? Maybe Paolo can answer this question. Are female with the familiar hypercholesterolemia have higher levels of LDL than males, or this is not the case when it's homozygous? What do you see in clinics? We cannot hear you, sir.
Okay, well, I mean, there are very few people, to be honest. In my experience, I just saw one young lady and a man. Now, the issue is that maybe not so many. Also, it depends by the location of the mutation also, which can be different. So because people can have 100%, both alleles can be affected, but the let's say the residual activity of the receptor can be from 0% to 10. I mean, it's a bit more. So I don't know if there are enough. This is a good point anyway. I don't have an answer.
Okay.
Maybe there is in the literature, but this is a point that has not been addressed probably that much.
Okay, a question from Adriana. Thank you for interesting presentation. I would like to ask you whether we still focus on plaque stability when investigating therapeutic approaches for atherosclerosis.
Yes, absolutely, but we don't have the model to do that. Unfortunately, this is the problem. Absolutely, plaque stability is very, very important.
Yeah.
Because you can have, let's say, not so extended atherosclerosis, but unstable plaques, which is very, very dangerous for thrombosis. So drugs that, let's say, target unstable plaques will be very, very critical. But unfortunately, we don't have models to do that. To let's say, to target unstable plaques and the methods to detect them, actually. This is a real problem in atherosclerosis. We still do not have methods, good methods to detect unstable plaques.
Especially in mice when you cannot go into the small arteries in heart and so on.
Exactly.
So.
Especially in mice. I mean, to try the drugs.
Yeah. Okay.
I have a comment and a question. I've seen that. I'm sorry, but it was so interesting. In most models, we saw stable plaques and always smooth muscle cells migrated from the media, right?
Yes. Yes.
So, which is a well-known association. So more collagen, more fibrous cap, and so on.
Yes.
So indeed, as you say, none of them is a good model for checking the effectiveness of drugs that are stabilizing the plaque, because these are already stable. On the contrary, the ApoE fibrillin knockout is kind of so much disrupted that maybe we cannot even think about.
That's true. That's very true.
Something in between we need.
Exactly. Which is not present, actually. We don't have it.
Yeah.
Okay, I'm seeing Johannes, he raised his hand. I don't know if you want another.
He wants to make a comment?
Yeah, maybe to. Okay.
Yes, it was actually on the inflammation model. I can just specify that. So we have a mouse strain that expresses constitutive active IKK2 as the main activator of NF-κB downstream of a LOXP flanked stop cassette. And we crossed that mice with a BMX-3 strain, inducible one, which expresses only in arterial endothelial cells. And by that, we could actually activate inflammation, persistent inflammation, only in arterial endothelial cells. We published that in atherosclerosis, and what we saw is that atherosclerosis was aggravated and accelerated. And furthermore, what we found is that we could then see that it drives with RNA-seq analysis and so on, that it drives the conversion of smooth muscle cells from the contractile to the synthetic and furthermore to the macrophage-like phenotype. So we could also see that the cells start losing their identity, so to say. And we also have a type 2 CRE-ERT2 mouse strain, so we can also activate inflammation in all endothelial cells, so in venous and arterial ones. But we didn't investigate that yet, so we want to start with it soon. So we don't know what the phenotype of that will be.
Excellent. Yeah. Thank you very much.
Okay, so we can conclude this webinar. I think the discussion was very nice, very interesting. Dimitris, thank you so much for giving this comprehensive overview and raising a lot of questions for the future to be answered. I just remind everybody that the next webinar is going to be on the 20th of December before the holidays, so we can all go and enjoy the holidays. It's going to be on next generation sequencing by Rosiane from Malta. She will give the lecture on use of next generation sequencing for genomics mostly. I wish you all nice week. Thank you so much for being here, and see you soon.
Thank you very much.
Bye-bye.
Thank you so much. Bye-bye. Thank you, Dimitris.
Bye-bye.